talk about three different groups of viruses. But these viruses can also be grouped into two based on the source of infection to human beings. So we're going to talk about rodent-borne and bat-borne viruses. As you may have known by now, most viruses, humans are not their natural hosts. They do reside in other animals, and humans do happen to be accidental hosts because of the interaction between humans and those animals. So today we're going to look at viruses which are mainly transmitted. They normally live in rats or rodents, where they sometimes don't cause disease, but when they cross over to human beings, they can cause very severe diseases. We're also going to look at a number of viruses also, 2 or 3 viruses, which also normally reside in bats, but they can be transmitted to human beings being accidental hosts. And so, these viruses that we're going to talk about today, bat-borne and rodent-borne, I say they are actually can be found in 3 families of viruses. The bat-borne viruses that we're going to talk about today are found in a family of viruses called Filoviridae. And we're going to look at one example of these viruses today. And then we're also going to look at rodent-borne a virus, which is in the family of Borne. And then finally, we shall look at another rodent-borne virus, which is in the Arenaviridae family. And so it's important to note at the beginning that, again, all these viruses that we're going to look at are purely topic. Done. Topic is rodent and bat-borne viruses. Is that clear? Good. So let's take a look at some of these viruses. We start with the family of Borneviridae, whereby we're going to look at within the family of Borneviridae, we have other viruses. I'm not sure whether we talked about these other viruses before. I think we did when we were talking about arthropod-borne viruses. We talked about CCHF and we talked about RVF. Do you remember what CCHF stands for? Do you remember what CCHF stands for? It's a virus that is transmitted to human beings. Can I see from the chart? What does CCHF stand for? We talked about this in the previous lecture, correct? CCHF stands for Crimea Congo hemorrhagic fever virus. Crimea Congo hemorrhagic fever virus. We may not have looked at it in details, but this virus is one of those viruses that are transmitted by ticks to human beings. And the name Crimea Congo was based on the fact that this virus was simultaneously discovered in a country which then was called Crimea, but has over the years been taken by Russia as part of Russia. And at the same time, when it was discovered in Crimea, it was also discovered in Congo. And then RVF stands for Rift Valley fever virus, a virus that is very common in our locality. So within this family of Borneviridae, we have viruses which are transmitted by arthropod, that is vectors, mosquitoes, and ticks, and these are CCHF and RVF. But we also have viruses which are transmitted to humans via rats or rodents, and this is going to be the focus of our lecture today. So we're going to look at hantaviruses. So take a note of the picture on this slide. That is how the viruses in the family of Bunyavridae look like under electron microscope. That is how they look like. This kind of a picture can be brought to you during practicals, so that you remember this. So I want us to take a little bit look at hantaviruses as one of those viruses of our focus of discussion today. Again, looking at, in terms of classification, we have said that these hantaviruses belong to the family of Bunyavridae in the genus hantaviruses. The viruses, there are about 20 species of hantaviruses. All of them, live untransmitted they are transmitted by rodents to human beings. And the interesting thing is that despite the fact that, they are different, they're classified in different species, they do share certain antigenic structure. They have what is called group and antigenicity. So that's a unique thing about these viruses. The other thing to note about the hantaviruses is that, out of the 20 known species of hantaviruses, they cause different types of diseases in humans. One group of disease they cause is called hemorrhagic fever with renal syndrome. Hemorrhagic fever with renal syndrome, or HFRS, hemorrhagic fever with renal syndrome. We're going to look at which hantaviruses cause this in detail later on. The other disease they cause is called hantavirus pulmonary syndrome. Hantavirus pulmonary syndrome. Again, we're going to look at which hantaviruses do actually cause this particular disease. So looking at hemorrhagic fever with renal syndrome, disease caused by some hantaviruses. You can see that on this table, it shows you the name of the virus. We have Dobrava, Sol, Pneumala. These are some of the serotypes. We also have Antana, some of the serotypes of hantaviruses that can cause hemorrhagic fever with renal syndrome. And on the same table, you can see the host. When you talk about rats or mice, there are also different species of mice that transmit these different viruses. And you can see that different hantaviruses are transmitted by different species of rats. Again, the table is showing you where exactly are these viruses domiciled. You can see, for example, Antana is mainly found in Asia, Far East, and Russia. If you look at, a virus like Sol is found worldwide. We are going to look at the epidemiology of viruses later, but then Pneumala is found in Europe, Dobrava is found in Europe and Balkanas. So you can start wondering, we're not seeing Africa here, but we shall talk about why we're not seeing Africa as one of the continents where these hantaviruses are common. So when the other disease that we talked about earlier that is caused by hantaviruses is called hantavirus pulmonary syndrome. Hantavirus pulmonary syndrome. And again, as I said earlier, different species of hantaviruses cause different diseases. And you can see that this hantavirus pulmonary syndrome is caused by examples of hantaviruses are Cynombre. Cynombre is mainly spread by deer mouse. Is very common in Central and Western America and in Canada. Then we have, Monongahela, hantavirus, which is spread by another species of mice, which is called P. maniculatus, and again is mainly common in the US and Canada. Majority of the viruses that cause hantavirus hantavirus pulmonary syndrome are mainly found in Canada and US, are mainly found in Canada and the United States of America. And so generally, when we look at the epidemiology of hantaviruses, we are told that hantaviruses are found worldwide, but where you find the hantavirus depends on where the rat, the host, the natural host of the virus lives. You have seen in my previous slide when I was talking about different species of hantaviruses, and you saw that they were also being spread by different rats. Those rats are usually located, their locality is in the area where they transmit the virus. So whereas you find hantaviruses all over the world, but then majority of them are mainly confined to Asia and North America, that is US and Canada, and also in Europe. One of the things also to remember is that you can see hantavirus with pulmonary syndrome is mainly found in North America, Central America, and South America. But mainly US carries the heaviest burden of this virus. Hemorrhagic fever, hemorrhagic fever with renal syndrome caused by hantaviruses is mainly, this disease is mainly confined in Europe and Asia. So if we say that these viruses are found worldwide, how comes we're not giving examples of Africa? In Africa, you know that there are rats, and some of us live with rats in our houses or wherever we live, we see them around. There has been studies which have done in humans about screening antibodies to hantaviruses, and we have found that quite a number of these hantaviruses, human beings have antibodies to them, but there has never been an outbreak of hantavirus. And so there is evidence that human beings living in Africa have also been exposed to hantavirus. But the two diseases that we're talking about today, that is hemorrhagic fever with renal syndrome and hantavirus with the pulmonary syndrome, are not common in Africa. So as a medical doctor, when you graduate, become a doctor, it will be very rare for you to find patients presenting with these symptoms, but you never know. The globe is changing. Maybe in future, we shall be seeing more of these diseases in Africa. But for now, they're only confined in the US, North America rather, US, Canada, Europe, and also in Asia. So transmission of hantaviruses. So hantaviruses are mainly spread via aerosols or bites. So when rats which are infected with this virus, so we're talking about transmission within rodents, and not the first transmission is within rodents. How do rats transmit the virus among themselves? So this transmission can occur either by them, breathing in or touching or biting each other. So if one is infected, they can transmit the virus through bites, or they can transmit the virus through, sneezing, through aerosol bite. But the uninfected rat can also acquire hantavirus through saliva from an infected rat, and also through feces, from an infected rat, or urine as well. How about human beings? How do we actually get infected? Again, it is important to note that whereas hantaviruses are mainly found in rodents, human beings are not a natural host. We are accidental host, which means we get infected when we come in contact with infected rodents or their droppings. So if you live in a house where there are rodents or rats, for that matter, sometimes they leave their, feces. They are very tiny portions, which you may not, surely see them. You may get in contact, put in your mouth, swallow, and then you get infected. So the transmission from rodents to human is mainly through contact with an infected mice or droplets from that particular mice or mouse. So then when you get infected, what happens? So we talked about two diseases caused by hantaviruses. The first one being hemorrhagic fever with Raynaud's syndrome. So the incubation is about 1 to 6 weeks. So the virus is replicating in your body, no symptoms yet. Then, after 6 weeks, then you start developing symptoms. That is hemorrhagic fever with Raynaud's syndrome caused by hantaviruses. The other disease that we talked about earlier caused by hantaviruses is hantavirus pulmonary syndrome. You can see that the incubation period is again 1 to how many weeks here? So we talk about 10 weeks, 1 to 10 weeks, or 1 to 7 weeks. 8 weeks, depending on the virus. So each of the viruses that constitute hantavirus, they do have different incubation, but we also will see later on that these viruses also have different severity when they infect those who are infected. So that's something that we may need to take note of. And so hemorrhagic fever with the Raynaud's syndrome. So again, this is one of the diseases caused by hantaviruses. Remember, we listed a number of viruses earlier, where we said that they cause this disease called hemorrhagic fever with Raynaud's syndrome. But again, we are told that out of those viruses, the severity varies from one virus to another. But the disease, come in stages, with severe disease showing up with febrile. You start feeling fever, general body weakness, you develop hypotensive, you start, the amount of urine that you produce is really reduced. You have a condition called oligonuria or diuretic, and then you start convulsing. And then, this may lead to death, or you may recover, depending on how your body is good in fighting these diseases. So I want you to go and, in your own time, read about the actual symptoms that occurs during the initial stages of the hemorrhagic fever with Raynaud's syndrome caused by hantaviruses. So the other disease caused by hantaviruses is the hantavirus pulmonary syndrome. It also occurs in 2 stages. We have the initial phase, which presents with fever, myalgia, and headache. And then, some people recover after that. Others do not recover. They move on to the second stage of the disease, which is called cardiopulmonary phase. And during this stage, you develop a bit of abrupt respiratory distress. You start having difficulties in breathing. You start, coughing, and develop pulmonary edema, and eventually develop cardiac, abnormalities. And again, this can also lead to death. Some people recover, some people die, depending on how likely you are. And so that is what I wanted to talk about hantaviruses. I always believe that mine is to do introduction. You probably go and read more about hantaviruses at your own time. Please focus on what we've talked about today, plus diagnosis and preventative methods. Diagnosis, preventative methods, and treatment. We have not covered that, but you please read that at your own time. We will not cover in this lecture. We will not cover arena viridae, which are also spread by rodents. I wanted to pick one disease spread by rodents, and then I leave you to read the others. But I'll share with you notes on this that you can read at your own leisure. And under Arena viridae, you shall be looking at Lassa, fever virus. You also read about Junin fever virus and Machupo fever virus. These viruses, they were given the names based on the places where they were discovered. So I'll share with you notes on this particular subtopic. The class rep, please remind me to send you the notes that you can read and understand more about rodent-borne viruses caused by the Arena viridae family of viruses. Allow me to dive into the bat-borne viruses. I'm sure most of you have kind of interacted with bats. Bats are all over, in cities, in villages, they live with us. And bats are proved to be one of the animals, rather, which can actually accommodate viruses without viruses causing disease in them. But those viruses, when they jump into human beings, they're likely to cause severe disease and even death. So today we're going to look at one of the viruses, actually two of the viruses, which are, their natural host are bats, but they interchange the natural host between bats and non-human primates. So we're going to look at the family of Filoviridae, which has two viruses. The viruses, one of them is very common, Ebola virus. The other virus you may or may not have heard about is called Marburg virus. These viruses, typically, if you look at them under electron microscope, they look like the picture that you see on my slide. They are filamentous. They are long, stretch, like filament. Yeah. So that's a very common characteristics of the Filoviridae. They are envelope viruses, and they are RNA. They are single-stranded RNA viruses. So that is something that you may need to, understand about these two viruses, the Ebola viruses and the Marburg viruses, which are, their natural host is thought to be bats that transmit them to human beings. And so I just wanted to say something about Marburg virus, but we will not, on my slide, you will not have any notes on Marburg virus. Marburg virus was first discovered in 1967. I'm sure most of you had not been born way earlier. In the early 60s and 70s, the vaccines that we used to use then, we used to, like the polio vaccine, which was used in those days, the vaccines were all obtained through growing them using monkey kidney cells. So people from Europe would come to Africa to look for monkeys, kill the monkeys, get the kidneys, and transport them to, actually they would get the monkeys and transport them to their country. So in 1967, there was a massive death of a number of people working in the lab and their families in two places. One is in Marburg in Germany, that's how the name came about, and Belgrade in Yugoslavia. And so the disease started from, the scientists who had collected green monkey from DRC to go and use the kidney cells from those monkeys to make polio vaccines. But then in the process, they got infected. They started developing what is called hemorrhagic fever disease. And so fever and then bleeding. And at that time, they took blood from these individuals and they were tested. And when the samples were looked at under electron microscope, what the virus they saw was very similar to Ebola, although it was not Ebola. So the virus was named as Marburg virus because it was first discovered in Marburg in Germany. That was the first time this virus, show up in human beings. then later on, we've had 2 cases in Kenya. There has been certain cases in South Africa, West Africa, but in Kenya, we have had 2 cases of individuals who suffered from Marburg disease. The first case was in 1980 when a German engineer who had gone to camp at Mount Elgon, in Western Kenya, after a few days, he developed, severe fever, started bleeding all over the body, was flown to Nairobi Hospital, and the gentleman recovered, but infected the nurse and the doctor. And I think one of them died, either the nurse or the doctor died, and then it disappeared like that. 7 years later, in 1987, another young tourist from Denmark also went visiting Mount Elgon and got infected. He also suffered the same fate. He then also transmitted the virus to the people who were handling him at the Nairobi Hospital. Again, I think there was 1 death from the gentleman did not die himself, but the healthcare worker, one of the healthcare workers who handled him, died. So those are the 2 times that we have had of Marburg in Kenya. So I want you to know that this virus called Marburg, it is somewhere in Kenya because we have had 2 cases, but we have never had anything else other than those 2 cases. And so I want to leave it there and jump and talk about the other virus, which we all know about, which is Ebola, Ebola virus. So before we jump into this, because I'm sure most of you actually know Ebola. I can see Jeff Correll is asking somebody whether I'm talking about Elgon. So how many types of Ebola do we have? I just want to see from the chart. How many types of Ebola do we have? Can we name them? Based on the chart, because Ebola is not a new virus. Can I see the type of Ebola viruses that we have? Anybody knows the type of Ebolas? 4, name them. Sudan, correct. Any other Ebola type that you know? Zaire, correct. Any other Ebola virus that we know? Prof, sisi tusiingilie mambo ya watu wawili. Okay. Pole sana. I didn't know that you guys were having a side chat. I was just reading the chat. Corinne, sorry for that. So we have Ebola Reston, correct? Ebola, that's correct. So those are the Ebola viruses that we do have. So let us look at this virus a little bit in details. The Ebola virus, I think we've heard about it recently in the news. So what is Ebola virus? Ebola virus is the most lethal virus known to human beings. Now, why is this so? Up to 90% of those who get infected with the Ebola virus die. Do you know that, there is, if you get a bunch of people, put them in a room and expose them to any disease, not everyone will die. Do you know about that? Most people, you will find I want to give an example of a study which was done in, it was not a study, by the way, there was an accident in Germany a few years ago. There's a plant where, you will find that they are making TB vaccines, and as the TB vaccine is based on BCG, bovine tuberculosis bacteria. But as they were making the bacteria weak, some of the bacteria actually, instead of becoming weak, become very pathogenic. And where they were making this, there were fumes coming from the lab, which actually, was able to spread in the neighborhood. So a lot of people got exposed to TB, very dangerous TB bacteria. And so the bacteria was so dangerous that, scientists thought that anyone exposed to it would die, but not everybody died. So some people got very sick and died. Some got sick and recovered. Some got just mild TB disease. Others never got infected. So that is the truth about diseases and human beings. So if you think about all diseases known to human, infectious diseases, Ebola kills more people than any other disease if you get infected. So what we call case fatality. Case fatality means if 10 people are exposed to Ebola, about 9 of them actually may die of the Ebola disease, which is totally different from COVID-19 was about 1 point something, 0 point something to 1, percent, and COVID-19 killed a lot of people. And so Ebola is very, very dangerous. That is number one. Number two, so this virus, Ebola virus, it is good to some extent that for many years, since it was discovered in 1976, the outbreak of Ebola have only been occurring in Central Africa, Central and West Africa. And the reason why this has been happening is because of the natural host. The bats that we're going to talk about today and the non-human primates that are also contributing to the transmission, the gorillas, are mainly found in the Central Republic of Africa and West Africa. And so that's something that you need to know about Ebola virus. So I asked you earlier, which type of Ebola virus we have, and I'm sure this slide of mine is old, so it left one of the Ebola viruses which you guys correctly picked. But again, here I'm showing you that we have Ebola Zaire, Ebola Sudan, Ebola Ivory Coast, and Ebola Reston. So then we have other Ebola viruses as well that you guys had mentioned earlier. Of importance also to note is that Ebola Reston has not been associated with human disease. Ebola Reston has not been associated with human disease. So the only ones which have been associated with human disease are Ebola Zaire, Ebola Sudan, and Ebola Ivory Coast. So looking back at the history of this virus, I don't want to spend more time on this, is that this virus was first discovered in 1976, both in Sudan and Zaire, and that is how the name came about. So that's how we came with Ebola Zaire and Ebola Sudan. But subsequently, there has also been discovery of Ebola virus in other countries like Gabon, Ivory Coast, and that has given Ebola, additional naming. So now we have about 5 different versions of Ebola virus. So I think one thing I wanted to mention about the history of Ebola and the epidemiology of Ebola virus. Since Ebola virus was discovered in 1976, there has been frequent outbreaks of Ebola virus in Central Africa. This occurs like every 5 to 10 years, and then it actually, kills people, and then it disappears. But in 2012, 2014, I think 2014, 2012, 2014, that was the worst outbreak of Ebola in West Africa, where it killed over 11 million people. If you go and read about that outbreak of Ebola, you will find out that it started with, a single person being exposed to feces of a bat, and then, that person then spread it to relatives who got sick, went to the hospital, and they spread it to the hospital staff members, and the members of the hospital then spread it to the region, to the town, and the spread went wild, covering, I think, more than 3 countries in West Africa, something that had never happened before. And so the question of where does Ebola virus hide? So as I said, it comes out in like an outbreak, and then it disappears. So when it disappears, where is it hiding? Ebola virus, research which has been done has shown that Ebola virus hides in one of the species of bats called fruit bats. And, there's evidence, antibody evidence showing that, Ebola virus antibodies are present in these mammals. And for that reason, scientists believe that bats are the natural host of Ebola virus. And that's why today we're talking about bat-borne viral diseases. So Ebola and Marburg viruses are believed to be harbored in bats. But also, it's important to note that there are very many types of species of bats. Not all species of bats harbor Ebola, and that's probably why, a country like Kenya, we have never had an outbreak of Ebola virus. Which is the most recent outbreak of Ebola virus that you guys know? With the most recent Ebola outbreak that you had in Uganda, yeah. So there's an ongoing outbreak of Ebola in Uganda, which has killed quite a number of people, including a healthcare doctor from Tanzania who was handling a patient who got infected and died. And so it is, whereas Ebola has been mainly confined to Central Africa, we're now seeing more and more of the virus moving to East Africa. We have never heard of Ebola case in Kenya ever, but you never know, in future it may actually be here. And so transmission. So we are told that for you to acquire the Ebola virus, you must be through direct contact with blood, secretions, organ, and semen of infected persons. Now, before we reach here, I want us to take one step back. The transmission of Ebola normally starts from either human getting contact with feces of bats, which are infected with Ebola virus, or human getting in contact with blood of non-human primates like the chimpanzees and the gorillas. So if you get in contact with either feces or a bat that is carrying Ebola, then you get infected. The same with someone who gets in contact with blood of non-human primate which is having Ebola virus. So that is the first mode of transmission of the virus from the natural host to humans. Now, once a human gets infected, then they become sick. The virus, is produced in large quantities in blood and also in all secretions of the body, and therefore getting in touch with blood or secretion from the body of someone who is infected with the virus can be a source of transmission of Ebola from one person to another. Ebola can also be transmitted through sexual intercourse. Ebola virus has been isolated in semen of those who are sick. Now, so how do people get Ebola from non-human primates? You have to understand cultures of different communities to appreciate that the reason why Ebola is common in Central Africa. In Central Africa, there's what they call bushmeat. Bushmeat is a delicacy, just like samaki fish is a delicacy from where I come from. So people in West Africa, they catch monkeys and to eat as what they call bushmeat. So if in the case of an infected monkey with Ebola, when you are slaughtering it, if there is an exchange of blood between the monkey to yourself, then you get infected with the Ebola virus. And so transmission is through blood. So there's transmission from human to human. Most transmission actually occur in the hostile environment. Most transmission occur in the hostile environment. And so between someone who has appeared in the hospital sick from Ebola can easily transmit the virus to healthcare workers. Healthcare workers are the second level of the most at-risk individual to acquire Ebola virus. So what happens when you get infected? Number one, the incubation period is usually between 20 to 21 days. It is important to note that during this period, you cannot transmit the virus. The incubation period is between 20 to 21 days, and during that period, you cannot transmit the virus. After 21 days, you start by having fever, headache, develop flu-like symptoms, fatigue, internal and external bleeding, massive hemorrhage, and then you actually die. So as we said earlier, that Ebola kills up to 90%, between 50, 90% of those who are infected. But those who recover, there are those who recover from Ebola. The interesting thing is that recovery takes so long, and even if you recover, you still have some mental issues. That you can also spread the virus, for a while. When you are sick, after you've recovered from this disease. So remember that even if someone has been discharged from the hospital, they should still be kept. Apparently, the people who have recovered from Ebola are kept in the hospital for, I think, 2 to 3 weeks to ensure that they are unable to transmit the virus to other people. So diagnosis of Ebola is through taking blood specimen from individuals who are infected and doing what is called ELISA or PCR. But you cannot do this test in ordinary laboratories. The tests are mainly done in what we call high containment laboratories. These are laboratories where the person handling the specimen is protected from getting infected by the specimen they are handling. So we have what we call level 3 and level 4 facilities where these samples are handled. These labs are not common. There are a few in Kenya. Sometimes you hear during Ebola outbreak, samples were sent to CDC or KEMRI. These are the places where you have the high containment lab where these tests are done. So do we have cure and for Ebola and Marburg for that matter? No, there is no cure for Ebola. If you get infected, you are given supportive care, and you are actually isolated from the other patient to avoid you transmitting the virus to other patients and other healthcare workers. So there's no treatment for those who have Ebola disease, but in terms of control, we have a vaccine for Ebola. The vaccine that we have can only protect you against Ebola Zaire. It cannot protect you against Ebola, Sudan and other Ebola viruses. So recently, when we had outbreak of Ebola virus in Uganda, the virus that was circulating there was Ebola Sudan. We could not use the Ebola Zaire vaccine to vaccinate people, but currently there are studies which are ongoing in Uganda using that vaccine to see whether it can actually offer protection against Ebola Sudan. So this slide here is very important for you to understand. How do you protect yourself from getting Ebola? Remember, if someone gets Ebola for the first time, they become sick, they run to the hospital. Whom do they find there in the hospital? They will find you, they'll find the nurse, they will find the pharmacist, they'll find those porters, everyone who is working in the hospital environment. And remember, by the time somebody is coming to the hospital, they're already showing symptoms, and symptoms include diarrhea, vomiting, all that kind of stuff that releases fluid that can transmit the virus. So when a patient comes, when you suspect you have a patient suffering from Ebola, what do? Number one, you must put this patient in isolation. You must put the patient in isolation ward, and when in isolation ward, when you're handling the patient, you must wear gloves, and these gloves must be double gloves. You wear the first layer of glove, and then the second layer of gloves, and then when you are every cloth that you use there must be left in the room where this patient is. You must also wear Google. I think I have a picture here I'm going to show you. You must cover all your clothes with the proper gown, so that any any contact with any fluid from the patient does not end up in your street clothes. The street clothes are the clothes that you are wearing when you come from home. So this should not be, you should not, you make sure that you cover yourself, including the shoes that you are wearing. You must cover your shoes with something that, so that you don't carry any potentially infected material of Ebola home. That's number one. So all the linens from the people who are infected must be incinerated. They must be autoclaved and incinerated so that no one else has a chance to get in contact with them. Number two, Ebola can also be still transmitted from dead people. You remember during COVID, there was a time you saw people being buried in a very scary manner. That practice was borrowed from West Africa during Ebola outbreak, where they discovered that actually somebody who has died from Ebola can still transmit the virus from the body fluid. So if somebody dies of Ebola, they must not be washed. There are people who have traditions and customs that require that they have to really wash their dead ones before they are buried. You should not touch someone who has died out of Ebola virus. And finally, it's important to note that Ebola disease is a reportable disease. It must be in any suspecting case, you must report to the public health agencies in your country, whichever country you're working. Good. So I think we are done. This last slide is about vaccines, and I've mentioned that now we have a vaccine for Ebola, but that vaccine can only protect people against Ebola Zaire, not Ebola Sudan and other Ebola viruses. So I'm looking at the chart. So Kelsey is asking, if someone gets Ebola and recovers, so they now have permanent immunity? No, the immunity, they do have protective antibody, but how long the antibody lasts, nobody knows, Kelsey. But we've seen during outbreaks of Ebola, where people have recovered, blood has been taken from them, serum has been purified and given to people who are sick. But during Ebola outbreak, people are desperate, to treat those who are sick. So if you recover, yes, at that point you have protection. But how long that protection lasts, we actually don't know. The second question, Christopher is asking this: so how long can mouse droppings carry antiviruses? The antiviruses can survive for a long time in the environment, so we want they can stay for a long. We don't know how long, but for a long time. No one has ever watched from the time the mouse drops the feces until the antivirus dies. But what we know is that they are very versatile. They can stay in the environment for a long time. So that is Kamunya is asking, that is passive. It won't last. Yes. So people, I said that attempts have been made to use serum or plasma from those who have recovered from Ebola to treat those who are sick with Ebola. But that is passive immunization. The antibodies wouldn't last long. Over time, they disappear, and you actually, remain free from Ebola antibodies. Prof, is it correct to say Ebola is an STI? STI means sexually transmitted infection. Yes, Ebola is sexually transmitted infection, but conventionally, Ebola are very rare cases. But as I said, Ebola is found in sperms, and those who are suffering from Ebola can transmit Ebola to those who are not infected through sexual intercourse. So after administration of the virus, I don't understand what you're asking. This is Kwamboka. After administration of the virus with the side effects of the vaccine, will the side effects of the vaccine be similar to the symptoms? I'm not sure what you're asking, but I believe that you're asking if someone is given the vaccine, that the vaccine induce side effects. Kwamboka, the current Ebola vaccine that we are using had been tested in human beings and it was found to be safe. And that is why it is currently recommended for use during Ebola outbreak. How long does Ebola stay in a dead person? We don't know. As long as the fluid is still there, we don't know. No one has ever kept a dead body just to figure out how long it stays. But we know for sure, just as immediately you die, while your body is still filled with fluid, if you touch the dead body, you can actually get infected with the Ebola virus. And with that, I want to take this opportunity to thank you very much for listening to my class. So the only lesson we are having, colleagues, I want to remind you that we have not covered is adenovirus. I don't know if we want to cover adenovirus and paramyxo. Did we cover that?
No.
Did we cover? Have we covered the adenovirus and paramyxoviruses? Not yet.
Not yet.
Not yet. So we are remaining with two lessons. We're remaining with adenovirus and paramyxoviruses and oncogenic viruses. So oncogenic viruses will be taught by Barclay. I will give my lecture on adenoviruses and paramyxoviruses. I think it's on the calendar, it is saying 2nd of November. So we shall look for a makeup class next week if you don't mind. Would that be okay?
Yes.
Good. So asanteni sana, enjoy your lunch. It is now 12:32. Have a good day and we'll see you again next time. Bye-bye.
Thank you.